I think you all know about how the DNA is sequenced, and DNA sequences nowadays has highly piling up due to the development of this next generation sequencing technology. Now, next generation sequencing technology is changing. It is going into third generation sequencing technology, where the read length, usually next generation sequencing technology, the base pair, the length of the DNA which we can sequence is around 150 base pair for Illumina. So, length of the DNA stretch we can sequence is 150 base pair for Illumina. So, Illumina is one of the most widely used machine in DNA next generation sequencing technology. So, in order to increase the read length, we have our genomic sequence, our DNA sequence is about 3 into 10 raised to 9 base pair. So, we don't have any sequencing technology to sequence from the first base to the 3 into 10 raised to 9 base continuously. So, I think you people which you are hearing may develop such a technology which will be more comfortable, so that we can start the sequencing from the first base and ends at the 3 into 10 raised to 9th base. So, that type of technology may develop in future. Now, what we do is that we cut the sequence into small fragments and sequence this all these fragments, and then we will assemble that fragments based on a reference genome which we already have in our database. So, we will assemble these fragments based on a reference genome which we already have. That is how we are doing the sequencing nowadays. So, nowadays the read length or the length of the fragment which we can sequence is here only the cutting length of the fragment which we can sequence by the latest next generation technology is 150 base pair only. So, we are now developing new the third generation sequencing technology which can sequence more than this, or which can increase the read length. So, we are developing more technologies like that nowadays. So, this is about the sequencing. We are entering to the sequencing. I am just not telling anything from the surface. I am going into the sequencing technology from the scratch only. So, we know that in 1869 we discovered the genetic material, that is a DNA, but it took I think 100 years, nearly 100 years to discover the structure of the DNA. It is not 100 years, it is 85 years something. So, in order to determine the structure of the DNA, it taken nearly 85 years more than that, and its chemical characterization is done in 1909, and the structure, you can see the structure and DNA discovery was about 85 years more than my calculation is not approximate only, I need to calculate. So, 1869 the DNA is discovered, and in 1953 the so I think around that so 1953 yes, it's around 84 years. 1963 the structure of the DNA is being solved by these great people, Watson and Crick. You all know, every biotechnologist know that. And after that about 20 years, nearly 25 years only, Sanger sequencing, in order to determine the sequence, the Sanger sequencing has invented. That type of Sanger sequencing was called as first generation sequencing. First generation sequencing, and Sanger with Sanger during that same year itself, Maxam-Gilbert also came with same nearby same year only. Maxam and Gilbert came with another chemical modification, another DNA sequencing which uses chemical modification, but that technology can't be used in a large scale while sequencing the genome, or can't be can be practically used this chemical modification technique. So these both of this technique, Sanger sequencing and Maxam-Gilbert sequencing, are referred to as the first generation sequencing technology. Where is my cursor? First generation sequencing technology. Okay. So after first generation sequencing technology, whatever sequencing technology new came are called next generation sequencing technology. So Sanger and Maxam-Gilbert sequencing technology is called first generation sequencing technology. And first genome to be sequenced is the bacteriophage 5X174. So this Sanger sequencing technology, but Maxam-Gilbert technology is not much practicably feasible. So it is usually by years its importance get lost, but we will be studying all these things. All these technologies which were developed through scientists, which need to be studied, because actually when we are studying next generation sequencing technology, we should study each and every, even it is outdated also, you should study the technology, because many things, many new ideas you will get from the technology, because these people has developed this sequencing technology by years of research. So if you study all this, you will get new ideas to proceed with it. So but still, Sanger sequencing technology is the base of all sequencing technology. Mainly the next generation sequencing technology is also somewhat, all these technologies are somewhat related to how Sanger sequencing has been done. So this sequencing technology was we can consider as the base of all sequencing technology now used until today. So first automated sequencing machine came at 1986. Then only the people got the thinking that we have the automated sequencing machine. Earlier the sequencing was done in a separate reaction tubes, and we used to load it in gel and find out the sequence. So the automated sequencing machine came in 1986, which changed the that sequencing scenario. So it's around my age only this whole sequencing technology have, but we have around 10 raised to 9 genomes, human genomes. We have seen in the initial slide, human genomes have been sequenced so far. I think human genome or genome, I need to check. Anyway, 10 raised to 9 genomes have been sequenced so far. It's human genome only. It's the 1000 genome project when we are saying only we have studied. 10 raised to 9 human genomes have been sequenced. I will recheck and tell which. Anyway, 10 raised to 9 genomes have been sequenced so far. These years. So that much the sequencing technology has been developed. So in 1990s, after the advent of this new automated machine, humans we have gone for the human genome project. So we started for sequencing the human genome, and it was government funded as well as the private company called Celera Genomics was involved. Both government and private funded project to us. And the 1992, the first sequencing factory at the Institute of Genomic Research, TIGR, got developed. So all sequencing method came after the first generation sequencing technology is called next generation sequencing. So we will be first studying the first generation sequencing technologies, and then we will go to next generation sequencing technology. And nowadays it is next generation, first generation all over. Nowadays the third generation sequencing technology is being evolved nowadays. So quick history of sequencing. In 1995, first bacterial genome, we have seen that first genome sequence is the 5X174. Then first bacterial genome sequence in 1995, that is Haemophilus influenza, and first animal genome of C. elegans, and human genome project is completed in 2003. It took 13 years to complete the whole genome. It started in 1990, but it took 13 years to complete, and it's 2.7 billion dollar is taken for this whole process. You need to think even around $1,000 or a bit more than that, we can now sequence our human genome. At that time it taken 13 years to complete, because the technology was not developed at that time. They used the Sanger sequencing and to sequence, and the technology was not much highly developed. There was no next generation sequencing machine at that time. So it taken 13 years and $2.7 billion to complete the human genome. So the completion of human genome means it's only the human genome is not completely sequenced until now. Also many of the sequences are still coming, because we have seen different assemblies in the UCSC genome browser. Yesterday I have said about the one assembly when we are selecting that particular gene. So different assemblies are still coming and coming. So we are just mostly, when you just go through a genome or just go through a gene, you can see many regions of the gene with what unconfirmed nucleotide N. They don't know what it is present in that region. So they are still sequencing and confirming what is present in that N region. So still the genome sequence is not complete. Now only recently only this Y chromosome is fully sequenced. So like that, many of the chromosomes are sequencing, and the sequencing and its updating is still going on. So we can't say that human genome is fully sequenced, but using these reference genomes only. 2003 also reference genome is available in the UCSC genome browser. Yeah, we have seen yesterday. So using this reference genomes only, we are assembling our sequence after the sequencing of our genome. Yesterday we used 2013's 13 assembly, and still 2021 assembly or something is there, but usually we use the 2013 assembly nowadays for the analysis. And 2005 the first next generation sequencing instrument came into. So 2005 the next generation sequencing machine came into. The first next generation sequencing machine was from Roche. Roche's 454 was the first machine. Where is my cursor? Yes. The first machine came was Roche company's 454. Usually this machine is outdated and is not used nowadays, but usually people who explain this next generation sequencing usually neglect this technology, first developed technology, but still I think it has some relevance, because many new technologies developed or many new technologies still use, such as ion torrent, all use the same principle, same way of the library preparation, all these things according to the Roche 454. So every technology has its own importance. So the first next generation sequencing instrument was found in 2005, and 2013 the greater than 10,000 genome sequence in the NCBI database. Now I have said about 10 raised to 9. In 2013 it's from the book only. 2013 it was 10,000 sequence only. Now we can, I think it's 40. Now it is 40 is changed into 9 zeros. That much DNA sequence has been developed in a decade. The 10 raised to 9 sequence has been developed after a decade. So we are into the sequencing technology. So I am just checking whether anyone is there to hear the class. So, the quick history of sequencing. So, the 1977, the first genome 5x174 was sequenced, and there are two types of first generation sequencing, that is the Sanger sequencing and Maxam-Gilbert sequencing. Sanger sequencing is called sequencing by synthesis, sequencing by synthesis, and the Maxam-Gilbert method is called sequencing by degradation. The chemical method is used in the Maxam-Gilbert, and Sanger sequence use DNA polymerase to sequence the genome, just like PCR. So, we are going to Maxam-Gilbert sequencing. The first sequencing method developed, we can't say this as first sequencing method, both of the Sanger and Maxam-Gilbert has been developed during the same period only. So, Maxam-Gilbert sequencing is a method of DNA sequencing developed by Alan Maxam and Walter Gilbert in 1976 to 77. So, these people have developed 76 77. At the same time only Sanger sequencing also developed. Both sequencing technology developed at the same time. This method based on nucleobase specific partial chemical modification of DNA. We modify the DNA chemically and cleaving the DNA using a chemical called piperidine of the DNA backbone at the site where modified nucleotides are there. We modify the bases and then we cleave at that site where modification is done using a chemical called piperidine. That is what we have done. And Maxam-Gilbert sequencing is the first widely adopted method for DNA sequencing. Yes, it is the first one. So, both developed the same year, but it's considered as the first DNA sequencing method. Okay. So, and along with the Sanger's dideoxy method represent the first generation DNA sequencing method. So, Sanger and Maxam-Gilbert sequencing technology is considered as the first generation sequencing technology. Every sequencing technology come after that is called next generation sequencing technology. Maxam-Gilbert sequencing is no longer widespread use. It's not much used because the practicality of the Maxam-Gilbert sequencing is very difficult. As the this is developed during 1977, is it? So, Carey Mullis has developed the PCR on 1983. So, as the PCR technology all got developed this much, so PCR polymerase based technology will be more feasible in the case of DNA DNA sequencing than a chemical method. So, the Maxam-Gilbert sequencing method is not widespread used today, have been supplanted by the next generation sequencing method. So, it is being taken over nowadays by the next generation sequencing technology. So, I hope you all studied these technologies. So, in the case of Maxam-Gilbert, we usually take 4 reaction tubes. We usually take 4 reaction tubes for our sequencing reaction. Then just follow 4 reaction tubes we will take. Maxam-Gilbert sequencing require radioactive labeling of 5' end of the DNA fragments to be sequenced and purification of the DNA. First step is that we label the radioactive phosphorus at the 5' end of the sequence to be the DNA to be sequenced. Okay. Second, the chemical treatment generates break at a small proportion of 1 or 2 of the 4 nucleotide base in each 4 reactions. So, we just taken 4 reaction tubes and just mark it as G, A plus G, C and T. Okay, we mark it as, first you label the or the DNA fragment to be sequenced at the 5' end with the radioactive phosphorus. Then you take 4 reaction tubes and label it with as G, A plus G, A plus G, C and T. So, sorry, C plus T, sorry. C and C plus T. Sorry. G, A plus G, C and C plus T. So, like that we will label. And then each tube we have a specific reactions, chemical reaction going to happen. The purines A plus G are depurinated using formic acid, okay, and guanines are methylated using dimethyl sulfate, and pyrimidines are going to be hydrolyzed using hydrazine, and addition of sodium chloride with the hydrazine inhibits the reaction with thiamine. It only gives C only reaction. This is what is happening here. So, nothing you need to think. We are just adding the radioactively labeled DNA into all the tubes. Its 5' end is radio labeled. Okay, I will label it with radioactive phosphate. Then we will add to the A plus G tube, we will add formic acid. Formic acid. Which will depurinate the formic acid will depurinate the purines A and G. We can see, we can just write our sequence A, T, G, C, T, G, C, T, G. So, these, when we add formic acid, this A, T and T side got depurinated. So, depurination occur in this A plus G tube. Second, the G only tube, G tube, what we add? We add dimethyl sulfate. The G tube, where is my, the G tube, we add dimethyl sulfate. This dimethyl sulfate, what it will do? It will methylate the G's only. Okay. So, not A plus T, sorry, A plus G. Sorry, I have marked depurination A plus G. You should have told me. So, I will just tell. Consider this is our sequence A, T, G, C, T, G, A, T, C, G. This is our sequence. Okay. So, in the A plus G tube, the formic acid depurinated A site, G site, G site, A site and G site. Okay. So, in the G tube, again I will write here A, T, G, C, T, G, A, T, C, G. So, in the G tube, dimethyl sulfate will methylate the G only. G is only. And in the C tube, what happen is that C tube we will add hydrazine plus sodium chloride. That is my cursor. Hydrazine we will add hydrazine plus sodium chloride NaCl, which will modify the C only. This C is only. This will modify the C is only. Hydrazine and this sodium chloride modifies C only. In the C plus T tube, what we will add is that we will add a hydrazine only. This hydrazine will modify C as well as T. Hydrazine will modify T as well as C. Okay. After modification of this, we will add a chemical called piperidine, hot piperidine, which can cleave at the position of the modified base. Wherever the base is modified, it can be cleaved at that position. So, we will cleave the DNA at the position of the modified base. So, we will get a series of labeled fragment is generated. So, we can see here. So, this is the sequence. We have 4 reaction tube. In A plus G tube, what will happen? A plus G tube we have added formic acid. It will modify the A and G. So, the piperidine will cut adjacent to that A. Okay. We can cut the sequence here. Okay. Which will give a sequence GCT, which can modify this G also. It can cut here, so it will not get any. We will get this modified phosphate only, radioactive phosphorus only. Here G can be modified, and piperidine can cut one of the fragment like this. So we can get a sequence, a fragment like this GCT AC. So randomly it will cut in different ways in different. Piperidine can cut the sequence in different ways in different fragments, which is different DNA. So A is modified, so it can cut here also. So we will get a fragment of GCT ACG ACGT. So like that 3 fragments we can get while cutting it with hot piperidine. The modification is done by formic acid. It forms depurination can occur. After the depurination, the piperidine can cut the DNA adjacent to that of the modification. Here the first modification, one of the DNA, the modification may occur in this G, so it can be cut adjacent to G, will not give any sequence with radioactive with having radioactivity. So another modification occur here in this A. So the piperidine can cut adjacent to the A, so that we will get a piece of DNA with the GCT. Another modification occur here, and piperidine will cut here. So, we will get a sequence of GCT AC, and another modification will occur here. So, we will get a cut with GCT ACGT. So, like that each sequencing reaction, in the case of G, what it will happen, their dimethyl sulfate will methylate the DNA only in G, so that only G, so that only G will be modified. Okay, only G will be modified. So, here we will not get again the first one sequence is usually missed in the Maxam-Gilbert sequencing. So, we can see G, it cannot be cut, it will not give any fragment. Another G is modified here, so that it will give like this PCT GCT AC. So, one fragment only it got. So, like that, see, in the C reaction it is treated with the treated with hydrazine and sodium chloride. So, the C only get modified. So, we will get G, C will get another C, where it is another C modification is there. So, GCTA, like that we will get fragment. So, like that we will get fragments, and we will run on the agarose gel. That fragments we will run on the agarose gel. We can see it here. We will run on the agarose gel. So, in this, in 4 wells, one well is marked A plus G, next well is marked G, next well is marked C, and next well is marked C plus T. So, these from each reaction tube we will load the content in the each wells, and A plus G give rise to 3 bands. So, you can number the number of nucleotide here. So, 3 bands, okay, you think about 3 bands, and G will give 1 band, because G have this one cut sequence only is there. So, C will give 2 bands, and C plus T will give 4 bands, you can see. Okay, how to get the sequence, that is what we are going to check now. So, we got the band which is at the lowermost position. We can look at the lowermost position. Okay. When we are looking at the lowermost position, we can see 2 bands, one at C and one at C plus T. So, C plus T also band is there, C also band is there. So, we if the band is at C also and C plus T also, it is not T, okay, it will be C only. When we come to next position, we can see a band at C plus T, but there is no band at C. So, it is not C. A band is at C plus T, as well as band is not there at C. So, it will be definitely T only. Then we come here at A plus G. A plus G, when we look, band is there. Then we look at G, there is no band. So, A plus G there is band, and G there is no band. So, next sequence will be A only. And then the fourth position, we just look at bands are there, C plus T as well as C. So, definitely C plus T as well as C it is there. So, definitely the sequence will be C. Next position, we can see the bands at A plus G and G. So, A plus G band is there, G also band is there. So, it will be G only. Next, we can see the band at C plus T, but not in C. There is no band in C. So, it will be definitely T only. And the last band, we usually find out the sequence from the lowermost band to the uppermost band. So, I missed telling that. So, the last sequence is A plus T there is band, G there is no band. It is definitely, it will be A. So, we are writing our sequence as a C. There it is. We can write it from the down to up. C T A C G T A. So, like what I said, the first sequence is can't be detected using these Maxam-Gilbert sequencing. So we can got CTA and CGTA. We got the sequence by Maxam-Gilbert sequencing of chemical modification sequencing method. This is how we calculate the or we find out the sequence using chemical modification method. Hope you understood. So anyway, I will again tell what it is. Chemical modification method. So this is how Maxam and Gilbert find out the DNA sequence. So the chemical modification is done in 3 different reaction tubes. People who are not understood. So we will do, this is our sequence. Actually we need to sequence this DNA. We are just in order to understand only, we are just showing it as a sequence so that you will understand. It is unknown DNA which we need to get sequenced. So we are just saying it with a sequence. Okay. So first we need to take this DNA and we need to radioactively label this DNA at the 5' end. By radioactive, this gel is usually done autoradiography so that we get these bands. So we are getting these bands by autoradiography only. So for detection purpose, we are labeling this DNA with radioactive phosphorus. Okay. Then what we do is that we will take this DNA in different reaction tubes. One reaction tube is labeled as A plus G, one will be labeled as G. One reaction tube will be labeled as C and one reaction tube will be labeled as C plus D. And in A plus G tube, we will add the DNA, this DNA, and add this DNA. So it will be having a a sequence like this, which we having a sequence like this. So this DNA is added to all the tubes, and then in the A plus G tube we add formic acid. Formic acid depurinate what A and G. So the A and G. Then we will add a hot piperidine chemical which can cut near to that chemical modification. Here the modification occur here G, A, G, A. These 4 position modification occur. So one cut, the piperidine, hot piperidine will cut here. So we will only get this radioactive phosphate. So we will not get any nucleotide here. So we this segment will be aborted. So next modification here occur is A. So the hot piperidine cut here. So we will get a fragment. It's randomly cut a different fragment. Okay. In one fragment itself 3 cuts can come. Okay. Actually it is randomly cutting the fragments, the DNA fragments which we have given. So one cut can occur in this adjacent to this modification. So it will give a 3 nucleotide fragment. You can see here. So then it can cut here also. It will give here G is modified. So it will give a 5 nucleotide fragment. And it can cut here also, piperidine and cut here also. So it will give a 7 nucleotide fragment. In the same way, in the G tube, we have added another chemical, another modification occur. Here we add the dimethyl sulfate, which methylate G only, not A. So their methylation occur. In G only, not A. A is not modified here. So A is not modified. I am removing that. So then we will add hot piperidine. So here also it will cut here. So we will not get any fragment. and it will cut here also, and we will get a fragment of 5 nucleotide GCTAC. And then C tube. In C tube, we add hydrazine plus sodium chloride. Hydrazine plus sodium chloride. I am just removing every dots. So, in C tube, we add hydrazine plus sodium chloride. So, it will modify the C and the C. Okay. Then the hot piperidine is added there, so that it will cut. Here one cut it will cut, and here one cut it will cut, and here cutting will give one nucleotide fragment, and here cutting it will give a 4 nucleotide fragment. Okay. In the last tube, what we will do? In the last tube, we will add a hydrazine only, not sodium chloride. Okay, we will add a hydrazine only. And hydrazine, what it will do? It will modify C as well as T. So, it can cut wherever it can cut. It can cut here one cut it can make 1 nucleotide fragment. Here one cut it can make a 2 nucleotide fragment. Here one cut it can make, we say, 4 nucleotide fragment. Here one cut it can make a 6 nucleotide fragment. So, all these are kept in a separate reaction tubes. Then we will take an agarose gel and load all these in the separate wells, A plus T, A plus G, G, C and T, C plus T. So, we will load. So, based on the size, the fragments are separated in the agarose gel. So, we will, after running the gel, we will get a lowermost band in C lane as well as CT lane. It is present, the C will be the sequence, or the same band is present as C plus T. So, this will definitely be C. You can see in the T band, the C plus T, the band is present, but it is not present in the C. So, it is not C, it will be T. Like that, we will define the third one. A plus T, the band is there. Okay, A plus G, it can be A or G, but in the case when we look at G, there is no band. So, it will be definitely A. So, C plus T band is there, and C the band is there. So, it will be definitely C. And then here A plus G band is there, G band is there. So, G band is there. We are first thinking that whether it is A or G, then G band is there. It is a confirmation. So, next sequence is G. The next is C plus T band is there, C there is no band. Definitely, if there is no band in the C, there is no confirmation of C. So, it will be T. And A plus G band is here, and G there is no band. So, G is not there. So, it will be A. Like this, we will find out the sequence from the lowermost portion of the gel to the uppermost portion, and we will write down like this, so that we will get the whole sequence. The first sequence we will not get, first nucleotide we will not get, rest of the sequence we will get by Maxam-Gilbert sequencing, the chemical modification sequencing. Hope you understood. So, we will continue with the next sequencing method, that is the Sanger sequencing. Is it okay? Getting? No response. Sanger sequencing also we will do the similar thing only, but the sequencing reactions I will do with the same thing, are a bit different. In Sanger sequencing, we will take 4 reaction tubes, just like Maxam-Gilbert, but we will label it as A, T, G, and C. So, we will label like this. Sanger sequencing is a sequencing by synthesis or the chain termination reaction occur in the Sanger sequencing. So, what we will do here? We will see it. So, Sanger sequencing uses a DNA polymerase for the sequencing reaction, and we will it uses a typical PCR reaction for the sequencing reaction, which uses only one primer, and for all 4 nucleotide plus 1 dideoxynucleotide is added to the each reaction mixture. The random termination at specific bases occur, and they are then separated by gel electrophoresis. That is what we are doing. So, in a reaction mixture, we will add the all dinucleotide. We in a PCR reaction we need water, we need enzyme, we need DNTPs, all these things we need. So, we will add all these things to the DNTPs, primers, water, enzyme, all these are the components of a PCR reaction. We add all the components to all the 4 tubes. Okay, all the components of the PCR reaction component, one primer plus DNTP plus water plus Mg2 plus ions plus enzyme, all we have added. Then we add our sequence. Which sequence? This sequence. We are taking this sequence only. Then we will add our gene, our DNA. That is this sequence only. We are add our DNA. Okay, our DNA to all the 4 tubes. Okay. Then we will add a special DNTP to all the tubes, special DNTP, that is called dideoxynucleotide, DDNTP. Dideoxynucleotide is the same thing they are showing, the 4 tubes marked with ATGC. We will add DNA polymerase, primer, all these things, and then we will add all DNTPs, ATP, GTP, CTP, TTP to all tubes, and then we will add a dideoxynucleotide, only one dideoxynucleotide to the one tube. Okay. So, this is what we are going to do. Where is dideoxynucleotide? Okay. When we see deoxynucleotide, it has a 3' OH end. A 3' OH end is required for the DNA polymerase to add the next nucleotide to the DNA. To adding the next nucleotide, a DNA polymerase require a 3' OH end. This 3' OH end is not seen in the dideoxynucleotide. So, during the DNA synthesis, if a dideoxynucleotide is incorporated to the growing chain, that chain growing will be terminated after adding that dideoxynucleotide. So, this is the principle behind the Sanger sequencing. Sanger sequencing uses a dideoxynucleotide to terminate the growing chain. So, it uses a typical PCR reaction which contains the PCR or the contents of the PCR mixture, plus each tube we will add a dideoxynucleotide. We will add every deoxynucleotide, every DNTP we will add at low concentration. We will add dideoxy ATP here in this tube. At a lower concentration, we will add dideoxy TTP in this tube. and dideoxy GTP in this tube and dideoxy CTP in this tube, and we are going for PCR amplification. Okay. So actually we have our sequence here GCT. We don't need any radioactive labeling or anything. ACG TA, this is our sequence. So our primer will be added here, and our DNA synthesis will be begin by the DNA polymerase. DNA polymerase will add the nucleotide C, okay, and then in the first reaction to what it happened, I am saying G, then it will add A, T, G, C, A, T. In some of the reactions, as the dideoxynucleotide A concentration is so low, it will synthesize the whole sequence without any addition of dideoxynucleotide. In some condition, sometimes the sum of the sequences in some of the amplifications, randomly dideoxynucleotide can be incorporated while synthesizing the DNA. So here at the A dideoxy A get incorporated. So the chain will get terminated here. After addition of dideoxy A, there is no 3' end for the DNA polymerase to continue the synthesis. So it will be terminated here. So then again the DNA polymerase synthesize another fragment from the template. So here the normal ATP got incorporated, there is no problem. So there is no chain termination occurred. When we added these A dideoxynucleotide got randomly incorporated, so the chain got terminated. So we will get a 2 nucleotide. We can write in the A2, we get a 2 nucleotide CGA and CGA TGCA. Okay, 2 nucleotide. So we have completed the A2. And just removing all this. What happened in A2? Removed that also. Okay. It's a complementary sequence. Usually in Sanger sequencing, we will get the complementary sequence only. We need to build up the actual sequence from the complementary sequence. Okay. So A2 is over. In T2, what it will occur? In T2, this is a complementary sequence, so I am just going with that. I removed this, I have rubbed this, erased this sequence, or else I will write. Okay, I will continue. So actually as this first it may synthesize properly without any addition of any dideoxy T nucleotide, but sometimes randomly dideoxy T may incorporate to the, it is randomly only, every time it may not be, every time it may be because the DNTPs are in higher concentration, so it usually incorporate more, but in some cases the dideoxynucleotide will incorporate, so that the chain will be terminated here, and sometimes the dideoxynucleotide is not incorporated here, it will continue synthesis up to here, and then a dideoxynucleotide get incorporated here. So in the T tube what we got? In the T tube what we got is that CGAT, CGAT, GCAT. So this is the sequence like this. So like this we need to go for G tube and C tube also. G tube and C tube. Go faster. So G tube and C tube also we need to. So actually G tube and C tube, as there is lot of deoxynucleotide, it may not terminate while synthesizing in usually, but it may terminate when if a dideoxynucleotide is incorporated. So in G tube it may terminate here, so it will get we will get a C only, or sorry, write it properly, C only. And another, it may continue synthesizing this G, then one dideoxynucleotide get incorporated here. Sorry, here it is 2 nucleotide, dideoxynucleotide is here, so it will be incorporated and it will be incorporated. So like this. So then the G tube it will be, where is this G tube? It will be CG plus a CG termination, CGATG. So G tube is done. Then we have the C tube. C tube, what will happen? Let me erase here. Let me erase this, erase this, erase this, erase this, erase. In C tube actually, as the DNTPs are very high, it may continue synthesizing in most cases, but some cases the C, when this was C incorporated itself, it can get terminated, or in some cases it will continue synthesizing A, T, G, then next C it will be terminated. So we can see in the C tube we have a C plus and C termination, C, G, A, T, G, C. So we have completed the reaction and we are going to run the gel. So we are going to run our agarose gel like this only. So I will mark like this gel. Okay, just listen. I don't know anyone is there left, so I am just explaining. I can't see whether people are there or not. Anyway, I need to explain. So I will take 4 wells. One well is marked with A, second well is marked with T, third well is marked with G, and fourth well is marked with C. So these things I am going to load, the contents I am going to load. A content I will just mark the number of, we will get a shorter fragment down and a larger fragment up. So how many yes, it is 7 I think. I will just mark 7, otherwise we can change it. 1, 2, 3, 4, 5, 6, 7. These are the regions where bands are going to come. In A we have a 3 nucleotide, okay. Our band will be coming in the 3 nucleotide region. And next we have at 7 nucleotide, is it? Count it. So 7 nucleotide. In A, one band we get the 7 nucleotide and one band we got at the 3rd nucleotide. In T we have at the 4 nucleotide region. In T we have at the 4 nucleotide region one band, and next is 8 nucleotide is there here. We need to find out 8 nucleotide. In the case of T, we have 8 nucleotide. Where it is? We will find out here. Actually, it is 8 nucleotide. We will find out 7 only in the case Maxam-Gilbert. In Sanger, we can find out all nucleotide. 8th nucleotide here. And in the G tube, 2 nucleotide region will have the first band. The G tube, 2 nucleotide region will have the first band, and the second band will be at 5 nucleotide region, 5, here 5, and the last C1 nucleotide region we have one band, and second band is at 6 nucleotide. Hope we got all the bands. So we will be deducing the band from here. The first nucleotide will be C, second nucleotide will be G, can look on. The third nucleotide will be A, the fourth nucleotide will be T, the fifth nucleotide will be G, the sixth nucleotide will be C, the seventh nucleotide will be A, and 8th nucleotide will be T. So we can write like this here. The sequence will be C, G, A, T, G, C, A, T. So this is the complementary sequence. So you can write the sequence G, C, T, A, C, G, T, A. Look at here, the same sequence. So like this, we can find out our sequence using Sanger's dideoxy sequencing technology. So this is how the Sanger sequencing technology work. Hope you understood. So the same example I have taken in both ways.